From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 7, recorded on March 22nd, 2016. Hi everybody, I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Greetings, Vincent. Great to be here.
Hello again, 1 month later, roughly, right?
That's right. We've got some pretty notable weather to report from the Salt Lake Valley. So I've been watching storms come across the valley all day. I think I might have mentioned before, we're sitting here, University of Utah has dropped into the foothills of the Wasatch Mountain front. Across the valley, about 20 miles away is the Ochre Front, and there have been rain and snowstorms just kind of pelting us on and off all day.
Wow. What's the temperature out there?
We're hovering right at about 0 C, maybe 1 or 2 C. Just this kind of rain and snow mix, and a slight potential disaster. My office started leaking. So I've got a garbage can up on one of my desks. But, as the old saying goes, the show must go on.
You bet. Well, when I woke up this morning and left, it was 0 C, but now it's 12 C.
Oh, wow.
Sunny, nice, bright, sunny sky here in New York City. So, we're in that funny time where it's cold in the morning and it warms up right away. So you're not quite sure what to wear.
Yep,
But,
we're doing that every day. We're also doing these sort of wild swings from spring into winter. And in fact, if I had to make a prediction, we haven't introduced him yet, but I think our guest today, you can find him this weekend with his 2 boys skiing in the mountains.
Yeah, you can still do skiing. Well, let's do that. We have a guest today, also from your department, Nels. Well, I guess it's not yours, but the same department you're in, the Department of Biology at the University of Utah, Mike Shapiro. Welcome to TWiEVO, Mike.
Thanks very much. Great to be here.
Let me say that again.
Sure.
Welcome to TWiEVO, Mike.
I have very little to say on the TWiEVO part.
I made a mistake.
So good to be on the Evo podcast.
Good for joining us. And Nels, you have an endless supply of terrific guests for our TWiEVO there over in Utah.
Yeah, I know it. It's getting a little bit regional, but I couldn't resist. So I think obviously we'll get into the details in a little bit on this beautiful paper that Mike's group just published in eLife. But there's really, fun and interesting evolutionary genetics going on here across campus. I recognize there's also fun and interesting evolutionary genetics going on across the country and around the world. And so, of course, as TWiEVO continues, we'll be sampling far and wide. But
just save a couple of them for when I visit.
Oh, of course. Yeah. We're looking forward to this TWiX podcast festival that's just in early works here that we'll be putting together for next year.
So what's the plan? We're going to do a TWiEVO and a TWiV and a TWIM. Can we do all three? Or can't?
Yeah, we could even throw in a quick TWIP if we wanted to. We've got a new Plasmodium guy who just got up and running.
Wow. That'd be quite an effort. I guess we'd have to spread it over 2 days or something like that.
Yeah, or maybe do some, kind of abbreviated half casts of each one, just sort of a all-star show or something. We'll see.
Yeah, it'd be fun.
Yeah.
Looking forward to it. All right, so Mike, we're going to talk about your paper, but I want to first find out a little bit about you and where you're from, Mike.
Originally, I'm from Los Angeles. I grew up in L.A., and went north for college at University of California, Berkeley. Go Bears. And then headed east for graduate school at Harvard Department of Organismic Evolutionary Biology. So I started my career actually in paleontology, not in developmental biology or genetics. So I've always sort of had a deep appreciation for biological diversity and patterns thereof. And then more recently, I've tried to move it more into mechanism than pattern. I then went back west to Stanford for a postdoc in the Department of Developmental Biology. And then almost 10 years ago now, I came here to the University of Utah. Been here ever since.
So Berkeley, did you enjoy that?
I absolutely loved it. Yeah.
I know a couple of virologists out there.
Uh-huh.
I don't I don't think you would know them. It's been a long time, and you probably didn't have any contact with them. But
yeah, and I haven't been a student there since 1994 either. So
yeah, right. I visited a couple of years ago. It was pretty nice. It's a nice, it's a nice place to be.
Beautiful campus. Yeah, beautiful area.
I remember the museum there with the dinosaur in the lobby. Right? Remember that? Yeah.
Actually, that building was closed during my entire undergraduate career, oddly enough. Wow. So where I worked as an undergraduate was in the Earth Sciences building, which is where the Paleontology Museum was.
Okay.
So I spent many hours in the basement or first floor preparing fossils.
So I think there was a T-Rex skeleton in there, and there was also a raptor of some kind hanging from...
there's a big T-Rex skeleton. There's, I think, a pterosaur hanging from the ceiling. Mm-hmm. There's a number of other...
Not a raptor, the flying guys, right?
Yeah. Big pterosaur.
And this is relevant for today's podcast, too.
Yeah, absolutely.
That's right.
About that.
Yep.
So
foreshadowing.
When you went to Harvard, who did you work with there?
I was officially in the laboratories of Fuzz Crompton and Ferris Jenkins, who are both vertebrate paleontologists. They're both interested in early mammals and late Triassic biodiversity. So I was technically working with them, and they very kindly allowed me to wander a bit. So after my second year of graduate school, I took the embryology course at Woods Hole, the Woods Hole Marine Biological Lab, which really sparked my interest in developmental biology and changed the course of my career entirely. So after my experience there, one of the faculty members very kindly agreed to take me under her wing and teach me some more developmental biology in her lab. And her name is Nadia Rosenthal, and that was at Mass General Hospital. And then I spent a couple of years working there and developed my thesis project on the evolution of limb reduction in Australian lizards, which let me go down to Australia every cold Boston winter to collect skinks on beaches and wineries and other fun places. And then a couple of years after that, I segued into Jim Hankins' lab when he arrived at Harvard from Boulder. So I spent a bit of time in a number of different labs trying to cobble together a semblance of a thesis project and acquire skills from a number of different types of biological research.
And your postdoc again was where? I forgot already.
That was at Stanford with David Kingsley,
and
Department of Developmental Biology.
What was that on?
So in my postdoc, that was really some of my first experience in genetics. And the reason I went to David's lab is that he had started working with a new model organism, the three-spined stickleback fish. And throughout their range in the northern hemisphere, these fish have really radical changes in a number of different morphological traits and behavior and physiology. And this was a great chance for me to understand a bit more about the genetic underpinnings of morphological variation. So when I was in his lab, I was primarily trying to understand the genetic basis of pelvic reduction in stickleback fish. We don't normally think of fish as having pelvises like we do, but just like we have hind limb outgrowths, they also have hind fin outgrowths. And in a number of different populations around the world, these populations of sticklebacks have completely lost these structures. So one of the tasks I had was to try to understand how that happened.
So, and Mike, when you came to Utah, then, if I'm remembering right, you really set up your lab as a stickleback kind of evolutionary genetics Evo-Devo lab. And then maybe can you tell us a little bit about this transition you made to pigeons and if it's still unfolding or how that's unfolding now?
I think it is still unfolding a bit. We were largely done with our stickleback work, at least at the bench. But it began unfolding, oddly enough, when I came to interview here at Utah. So one of my now colleagues was my host for my interview visit. I gave my job seminar on sticklebacks and said how amazingly diverse they are, and this is such a great system. And I completely believe that, of course. But the next morning after my talk, he came to pick me up for breakfast. And in his backpack was this massive volume. And he pulled it out of his backpack and he said, those sticklebacks are pretty cool, but have you seen this? And he just plunked down this 800-page encyclopedia of pigeon breeds. And I was completely blown away. I mean, I knew a little bit about pigeons, and I knew the importance of pigeons to Darwin, for example, in trying to communicate his ideas about how selection works. But I really had no idea they were as diverse as they are. So the seed was really planted at that time to start thinking about other models of biological diversity and potential genetic models. I think that was the key. There are lots and lots of highly diverse organisms out there, but rarely do you see the level of diversity that approaches the level that you see usually between species, but within a single species. And what that allows us to do is do traditional genetic studies, to do crosses in the laboratory, to do whole genome sequencing now with the expectation that different representatives within the species are going to be fairly similar, with the exception of certain places throughout the genome that are controlling the differences that we're interested in.
Yeah, that's cool. So do you think that this pigeon book actually had a role in bringing you to Utah?
Well, I will say that what I saw when I visited Utah was an attitude, a philosophy of really going a little bit outside the box and thinking about unusual projects. And the fact that I got this vibe even during my visit that Utah would be receptive to doing something totally different than I was doing at the time and that I was trying to get this job with was actually pretty attractive. And the fact that there was potentially space to do it. And it really I didn't really go anywhere with it for a couple years after I arrived. And I think Nels knows this story, but I went to the state fair, which is out near the airport in Salt Lake City. And near the cow barn and the sheep pen and the rabbit hutch and any other variety of agricultural animals and pets on display, there was a pigeon shed. And it was manned by a couple of old timers from the local pigeon club. And seeing these things in person just absolutely blew me away. I mean, these things were like space aliens, all the different feathers coming out of every which place. Lots of differences in size. They differ in size by over an order of magnitude among different breeds. And I was just absolutely captivated. And this seemed like a great opportunity to do something similar to what we had been doing in sticklebacks, which was to do quantitative trait locus mapping. So I approached the guys who were meeting visitors in this shed and introduced myself and explained a bit about what I wanted to do. I said, I want to take that breed over there with the giant beak and the feathers sticking out of its feet, and it's twice as big as that one over there. And I want to cross it to that other breed that's totally different from it, cross the kids together, and then in that second generation, if the traits are not incredibly complex, what we'd expect to see is some of these traits segregating in the F2s. And these pigeon breeders are amazing geneticists, classical Mendelian geneticists. They totally get it. So when I explain this scenario, they're with me the whole time, like, oh, yeah, that's great. That'll work. But, you're going to have all those F2s. You're proposing to make hundreds of F2s. They're going to be hybrids, right? You can't show those anymore. They don't meet the breed standards. So there was this little gap in communication. I tried explaining it again. They said, oh, no, we get it. That'll totally work. That's a great idea. But, you're going to have hundreds of F2s and you can't show them. I don't know what you're going to do with them all. So eventually I was able to contact somebody else in the club who really had been waiting for somebody to do this for years and years. And once we had him sort of on our side, he was able to give us the street cred to move forward and sample people's prize birds without fear that we would exsanguinate them and bleed them on the spot.
So this diversity, is this because of the breeders or do they exist in nature?
This is almost exclusively from breeders doing selective breeding over the past approximately 5,000 years.
Wow.
Pigeons were domesticated quite a while ago. There's evidence, written evidence from Egypt and elsewhere from about 3000 BC that pigeons were being used for ceremonial purposes and for food, which is probably where some of these disgustingly enormous breeds came from. And then if you look back in even illustrated books going back 400 or 500 years, they show pictures of these domesticated breeds, and they really already look like they do today. So some of these traits like feathered feet and big and small beaks and feather crests and other different types of traits, these have been around for quite a long time. And we're really just exploiting the selection that's happened at the hands of breeders over many, many centuries, if not millennia.
So I think you hinted at it, Mike, but this connection back to Darwin as well. How did pigeons get his attention?
Darwin really was immersed in the pigeon societies of his day, the peristeronic societies of London. That's your word for the day, peristeronic, pertaining to pigeons. And Darwin was fascinated by by breeders and the outcomes that they could achieve with selective breeding. So he was interested in pigeons, he was interested in dogs, and a number of other domesticated species as well. And he kept pigeons himself, and he did his own breeding experiments. So Darwin saw this as an opportunity to try to understand something about heredity, but also to communicate to his audience in Origin of the Species and elsewhere about how selection works. So there is a long, interesting history of pigeons in evolutionary biology.
So the paper that Nels alluded to, which is published in eLife, Molecular Shifts in Limb Identity Underlie Development of Feathered Feet in Two Domestic Avian Species. Is this the first pigeon paper you've done, or is it one of a series?
This is one of a series. So our very first publication on this topic was, I think, in 2012, where we just wanted a basic understanding of the population genetics of the different breeds. How closely related were they? How genetically similar were they? Did we see the distribution of similar traits in different genetically related breeds? Genetically unrelated breeds. And the answer in large part was yes to that latter question, which was really encouraging for us because that gave us a clue that genetically unrelated breeds had the same traits, and it was possible they had those same traits because of introgression. So perhaps the same genetic variants or haplotypes were selected over and over again in different breeds. And what that allows us to do is identify those regions that the different breeds with a particular trait have in common against essentially a jumbled up genetic background. So using that, or hoping that that was the case, we proceeded to try to map a number of different traits, including head crests. So a lot of different breeds of domestic pigeon have a series of feathers that grow up on the back of their head instead of down. And they come in lots of different shapes and sizes as well. So I think about 3 years ago, we were able to map that trait in a bunch of different breeds to a single coding variant in a gene called ephrin receptor B2. Subsequent to that, we addressed some variation in color that's present among a lot of different breeds of pigeon. And we identified a small series of interactions among several genes that are well known to be responsible for color variation in other species. So we were able to explain about 8 different colors by examining interactions among loci and alleles of 3 different genes. And then this year, or just actually about last week, we published this paper on the genetic and developmental basis of feathered feet, which frankly for me was one of the sort of secret reasons for getting into pigeons in the first place. So my checkered past as a dinosaur paleontologist sort of came to the surface when thinking about this problem. Because one of the big changes that occurred between dinosaurs and their avian descendants was the transition from scales to feathers throughout the body. In most species of birds, there's very little variation in the placement of feathers and scales. Usually they've got a feathered body and then scaled feet, and in a small number of species there are feathers on the feet. But rarely do you see variation within a species, which prohibits any sort of genetic approach to understanding how that variation occurs. So pigeons are one of the very few examples of a species where there's actually variation within it. So that gave us an opportunity to take both traditional genetic mapping approaches as well as the genomics approaches that I discussed and try to understand what those feather-footed breeds have in common that the scale-footed breeds don't. And what we kind of half expected based on studies of chickens throughout the last couple of decades. So you can mess with chicken embryos pretty easily and misexpress genes, either by delivering them through viruses or electroporation or other methods. And in doing so, a number of different groups have shown that a number of different genes and pathways have the potential to change scaled epidermis into feathered epidermis. And so we half expected one of those pathways to pop up in our scan of the genomes of pigeons with feathered feet versus scaled feet. But it ended up being something completely different, which we can dig into further.
We'll get into that. But before you do that, you mentioned that, well, first of all, the dinosaur bird story. How do we know that birds are descended from dinosaurs? We don't have any dino DNA, right?
We don't really have any dino DNA, but we can see shared derived characteristics at the skeletal level that only birds and dinosaurs share, and nobody else has got them. So that case seems pretty solid to me, as well as I think most other evolutionary biologists who have thought about it.
Okay. And now, did any dinosaurs have feathers?
They did, yeah. So they had feathers on their forelimbs. They probably had feathers throughout their body, some of them. And we're talking primarily now about a group of dinosaurs called the Theropods, which are meat-eating dinosaurs. They encompass a huge number of species. The ones that we've got some better evidence of feathering for are the small theropods, the raptor-like theropods that were mentioned earlier. And some of them, including a particular dinosaur called Microraptor, actually had feathers on their feet, pretty long feathers on their feet. And not only were those feathers long, but they were also asymmetrical, which is very characteristic of flight feathers. So there's a central rachis or vein running up the middle of a feather. I'm sure everybody's familiar with that. There's barbs shooting out of either side of that central rachis. And in flight feathers, those barbs are longer on one side than they are on the other. And Microraptor, as well as some other feather-footed dinosaurs, had these asymmetrical-looking feathers on their feet.
So, Mike, maybe you can help us put this a little bit in perspective or the framework here. So the fact that feathers were near or on feet or hind limbs of dinosaurs so long ago, and yet when we're looking at these pigeons, it sounds like you're thinking about this in terms of them sort of picking this up very recently, sort of under the selection of breeders. Right. So what's sort of ancestral here, or how do you think about the relationship between these features, between these guys?
Yeah, that's a really interesting point. And I think what's emerging from the paleontological literature lately is that feathered feet may have been ancestral for birds. So early birds probably had completely feathered feet, and they weren't necessarily long feathers like we see in some of these pigeons, but they probably did have skin covered with feathers all the way down to their toes. So another way of thinking about this, which is I think what your question is getting at, is that modern birds may actually have secondarily derived scales on their feet. And in trying to understand what is putting feathers on the feet of pigeons, we may, in fact, be understanding a little bit about what got rid of those feathers in the first place, although we don't know that for sure at this point.
Do we understand? So when I think of dinosaurs, I think of rather large animals, most of which don't fly. What was the reason? What was the pressure for reduction in size and acquisition of flight? Do we understand that?
Ooh, I'm probably not the best person to ask this question right now. I mean, certainly some of the members of the theropods, a lot of them were quite small, even down to chicken size. Some of them were, it appears, feathered. I mean, you can imagine advantages of flight in acquiring new food sources, for example. That may not have necessarily been the original advantage for having feathers, though. I mean, certainly feathers are advantageous for thermal regulation. They provide some insulation. They can also be important for sexual display. Feathers are very colorful. So there may have been a number of different reasons other than flight that were advantageous when they first came about.
Boy, I bet people who study this would love to have some really old DNA, right?
Absolutely. So would we at this point. That's proven a bit elusive, as you might imagine.
Yeah, well, I don't think we... what's the oldest DNA? Neanderthal or something like that? That's not very old, right?
No, you can go back a bit farther than that. More. So the Denisovians are back. And I think people are pushing it even farther back, and maybe there's some potential that we will start to go, but I don't think we're anywhere close to the sort of Jurassic Park ideas of
So we're not even close to 1 million years. We're talking about a few 100,000 years.
At best. I think we're still under 100,000.
Yeah. Well, you never know what you'll find, right? You got to keep looking.
Yeah. And I think the big issue is degradation. Yeah, it's just the DNA just falls apart.
I don't know. So Nels, I don't know if not having DNA disqualifies people from TWiEVO, but it would be nice to have a dino person that could really talk about this, right?
Good idea. Well, and Mike in his ancient past, in his training, I mean, he has some of that. But I agree. It's a fun topic to consider and to get a view of what's going on in that field. I think that fits into our mission statement.
Right. Anyway, sorry for the digression. That's
no, it's okay. And it would be great if I could keep up with all these different bodies of literature. But as everybody knows, it gets harder and harder.
Well, when we find someone, maybe you want to join us just to.
Sure. Yeah, that'd be great.
Enjoy the conversation, and maybe you could advise Nels and I who to get at some point.
Okay.
Good idea.
Anyway, for today's story, why don't you tell us? So you're interested in how the difference in feathering versus scales on pigeon feet, and you have different pigeons that have one or the other. So tell us how you went about doing this.
Yeah, so there are many, many different breeds, both with and without feathers on their feet. And I don't remember if I mentioned earlier, there are about 350 different breeds of domestic pigeon. So there's quite a few of them. And previously...
I have to tell you, Mike, sorry to interrupt.
Sure.
When I think of pigeons, I'm here in New York City. You know what I think of?
Yeah, we're going to get you some nice pictures to go along with.
Some of these guys here, they don't even have feet.
Yeah. There are quite a few city pigeons who are missing toes and lots of other body parts, unfortunately.
That's not a genetic
No, we'll get you some pretty pictures to show you a bit more about the types of birds I'm talking about.
Okay. I think that's hard urban living.
Yeah, it's tough.
Yeah.
Tough to be an animal in New York. Anyway, sorry, Mike, go ahead.
No, it's okay. So early on in this process where we were interested in, as I mentioned before, studying the genetic basis of head crests and color variation and so forth, we wanted to start out with a group of genomes that could potentially address lots of different questions. So we sequenced about 40 different genomes of different breeds, and these breeds represented lots of different traits. Again, color variation, feathers on the feet, crests on the head, big beak, small beak, and so on. For the study that was published last week, we binned these genomes or grouped them depending on their phenotype or traits, specifically with respect to the feathers on their feet. So we had, I think it was 30-some breed or 20-some breeds with scales on their feet, and about 15 breeds with feathers on their feet to one degree or another. And we simply scanned across the genome, asking where they had some pronounced differences in their DNA sequences.
Were these whole genomes or?
These were whole genomes. So there was one very well done reference genome. And subsequent to that, we sequenced a lot of other genomes at much lower coverage. So we could do it a lot more cheaply after that one really good genome was assembled.
What's the size of the pigeon genome?
It's about 1.2 or so gigabases, which is on par with just about every other bird. Okay. Birds appear to be really quite conservative in a lot of ways in their genomes, including size.
Is it true that hummingbirds have smaller genomes, or am I thinking? So I'm thinking. About something.
I don't remember. I don't think they're much smaller.
Okay.
Actually. So if you look more broadly at things that fly versus things that don't. So for example, bats have smaller genomes, I think, than most mammals. But among birds, there's actually not a ton of variation. When you go out to the birds that don't fly anymore, like ratites, their genomes are actually bigger, interestingly. So among the things that fly, the size of the genome appears to be pretty consistent. So one approach of trying to understand which parts of the genome were important for feathered feet, again, is we compared the ones with feathers to the ones without. And when we did that, we saw two regions of the genome pop up that were highly differentiated between those two groups. In parallel to that, we have a breeding program in my lab up on the roof of our building where we breed lots and lots of pigeons. And what we did is essentially what I described earlier to the pigeon breeders at the state fair back in 2007 or 8 or whenever that was. We got a breed of pigeon with big feathered feet and crossed it to another pigeon that had scaled feet, produced F1 offspring, then intercrossed those F1s. And in the F1s, they had these sort of pathetic mix of scales and feathers on their feet. But in the F2s...
Isn't that in the supplemental figures? You can see those sad feet
the pathetic F1 feet. Yeah, exactly. So that's not a real desirable trait for the breeders. So you don't see that much in real breeds.
And I guess there's where artificial selection has sort of an ugly face to it when you go behind the state fair barn and the undesirable pigeons are sort of culled from the flock, so to speak. Is that true?
That's exactly right. So, and it is no pun intended, but cutthroat, where even a few feathers out of place or feathers that are the wrong color will basically kill a bird's chances of winning a competition, and those birds are culled from the flock. So it's pretty hardcore. So yeah, you would never see a bird like that F1 that we show in the supplemental figure. That thing wouldn't exist. In the F2 generation, and we've got pictures of this in the supplemental data as well, or the figure supplement, whatever eLife calls it. There was quite a range of phenotypes. There were some birds that had almost no trace of feathers at all. There were some that had lightly feathered feet, some that were a little heavier, and then others had what the breeders call muffs, which are these massive feathers on the feet just shooting out everywhere. And what that told us right off the bat was that this probably wasn't a simple trait, meaning it wasn't a single gene controlling all this variation, because we saw enough variation in the F2s that it told us it probably wasn't just one locus or one gene controlling everything. But what we could also see is that it probably wasn't 100 different genes of equal effect. If that was the case, then all of the birds from that second generation would all pretty much look intermediate. They'd all kind of look alike. Mm-hmm. So we knew it was a modest number of genes that were involved. The advantage of having a cross in the laboratory is that we could then genotype those birds across the genome and see which parts of the genome they inherited either from the feather-footed parent or the scale-footed parent, and try to find associations between those patterns of inheritance and the phenotype that they had. Mm-hmm. And when we did that, we found that one place in the genome primarily controlled how much feathering there was on the feet. So was it just a little bit of the foot? Did it go all the way down to the tips of the toes, or what? And then a second place in the genome controlled how long those feathers were. So two different loci, or two different parts of the genome that were doing slightly different things. And it turned out those two places that we found through those laboratory crosses were the exact same places that we found by sequencing the whole genomes. Mm. So that was really encouraging. Two completely independent approaches had converged basically on the exact two same places in the genome. So that was very encouraging. With the laboratory crosses, just because we can't generate thousands and thousands of offspring, the region that we knew was involved in this trait was large. It was probably several megabases. It was very big, and there are hundreds of genes in that region. On the other hand, the region that we identified through the whole genome sequencing was considerably smaller, and that gave us some clues about which genes were actually involved.
So, Mike, how many offspring are we talking about when you say you can't make a lot?
For last week's paper, we genotyped and phenotyped, meaning we measured all the feathers and scales, or the extent of scales and feathers, in 130 offspring. Mm-hmm.
And Mike, when you're looking at these traits, so it's interesting in the paper how you break it. It looks like you break it down to like single digits or single toes, I guess. Right. As you're sort of making these measurements and assessments. Do you really have to go to that level of detail, or what do you gain by doing it at that sort of a fine-grained view?
Yeah, what we were really interested in is trying to understand where these feathers were growing out. Was it just sort of a general feathering of the feet? Wherever there used to be scales, there are now feathers? Or was it a little bit more specific than that? And what we found by treating the foot on a digit-by-digit basis was that the feathers, especially the big ones, were primarily coming out of the lateral digits, the ones that were more towards the outside, where our pinky toe would be, for example. So that's very different than feathers just shooting out everywhere. They were primarily coming out of the posterior or lateral digits and facing outward.
So now you had narrowed it down to a certain region of the genome, but it's still pretty big, right?
It wasn't too big. I mean, we're talking about dozens or a couple hundred kilobases rather than megabases. Mm-hmm. And in a vertebrate genome, that narrows the number of genes down quite a bit. Right. So at that point, we looked at these regions of high differentiation between the scale-footed and feather-footed birds, and we didn't find any changes to the genes themselves that suggested the gene was broken. Right. Or had some other sort of altered function consistently in the birds that had feathered feet versus the ones that didn't. So that is different than what we found, for example.
Oh, I just want to know if there are any amino acid differences in the in the different feathers, without feathers, in these genes, in these loci, or were they absolutely identical?
I'm trying to remember. There were no consistent amino acid substitutions in one group versus the other. Yeah. So we now have a rather small number of genes that are in this region, in these two regions that were highly differentiated between the two. We didn't see any changes that screamed out to us as being that could alter protein function, for example. And that, again, is different from what we found previously, for example, with head crest. The head crest mutation appears to drastically alter the function of a receptor tyrosine kinase. In the case of the feathered feet, we didn't see that. So we then asked whether there were differences in the way some of the genes in these differentiated regions were being expressed. So one way to cause a change in the way development occurs is to change the function of the gene. But another way is to change when or where or how much a particular gene is being expressed. So we measured the level of gene expression of a number of genes in these differentiated regions. And for the most part, there weren't differences between birds that were either scaled or had small muffs or had really big muffs. But one gene under each of those peaks showed some pronounced differences in expression levels. Under one of the peaks, the expression level of the gene actually went down. And that turned out to be a gene called PITX1. It's a homeobox-containing transcription factor that's been implicated in, well, a number of different things, including hindlimb development and conferring identity to a hindlimb. So if you get rid of PITX1 genetically in a mouse, its hindlimbs are pretty screwed up. It turns out also, ironically, that it's really important in pelvic reduction in stickleback fish, which is what I studied as a postdoc.
So sort of convergent scientific evolution here.
Yeah, exactly. So I completely switched systems, and I still can't get away from this gene.
So, Mike, maybe let me ask, so you're kind of moving already from the QTL, or the quantitative trait locus mapping, into actually measuring expression to sort of narrow down to the sort of causative variation. How much did the kind of population genetics play in narrowing this? So, in the paper you mentioned this probability FST testing. Maybe could you fill that in a little bit?
Yeah, we're basically looking at differences in allele frequencies between feather-footed birds and non-feather-footed birds or scale-footed birds. So are there sequences in the feather-footed birds that are present often or always that are not present in the same place in the birds with scales? So that essentially is what the P, or probabilistic FST metric, told us, was were there significant differences in frequencies of alleles at that position. So it was actually incredibly informative to narrow down the regions of the genome that were actually differentiated and probably mattered in these in the evolution of these traits, or this trait.
Mm-hmm. And then getting back, you mentioned this amazing sort of history of the pigeons, the breeding programs and so forth. And so is it true that actually these mutations were known or described as the grouse phenotype and the slipper phenotype? And then it's sort of like you connected this in a modern way with this genomic approach?
That's right. These loci are well known and have been well known for quite some time. And sort of the golden age of pigeon genetics goes back about a century to a number of, well, at least in the pigeon community, very well-known pigeon geneticists, and some that are known outside the pigeon community too, such as T.H. Morgan, who was an avid pigeon breeder himself, believe it or not. So these grouse and slipper loci have been known for quite a while. And so the genetic architecture of this trait has been understood to some extent for quite a while as well. But the molecular basis of it, the molecules that are actually at these loci, were completely unknown. So as you said, we're trying to connect the dots between what's known from classical genetics and what we can get some traction with in molecular genetics using much more modern tools.
So PITX1, a transcription factor, when you saw that, did you get happy?
I had mixed feelings. I was actually hoping to find something a little different. As I mentioned, we were still working on sticklebacks at the time, and it would have been nice to see something a little different, perhaps.
Yeah.
But still, we knew a lot about the gene, and biologically it made a lot of sense. So we're talking here about a trait that really just affects the hind limb. Mm-hmm. It's just affecting the legs. And there are not that many genes that are only expressed in the leg. PIDX1 is one of them. There are a lot of other genes involved in limb development, but they tend to be involved in development of both the forelimb and the hind limb. So you can envision tweaks to some of those other really important genes could have some profound deleterious effects on development, either in both limbs or elsewhere as well. PIDX1, for example, itself has a number of different roles during development. It's not just involved in the limb. The name, or the pit part of the name, comes from the pituitary. It's also expressed in the pituitary. It's also expressed in the jaw. And the mouse knockouts that I alluded to earlier, they not only have messed up limbs, but they also have defective jaws, and brain development is affected as well. And those mice die shortly after birth. So mutations to PIDX1 to alter, to the gene itself, to the coding region, to alter aspects of hind limb morphology are kind of a bad way to alter your legs and live to tell about it. So these regulatory mutations, like we think we've identified here, circumvent those deleterious pleiotropic effects, those effects on other regions of the embryo that depend on proper activity and expression of things like PIDX1 for normal development.
Yeah, so this idea has come up a few times on TWiEVO now, which is sort of this, these observations as more and more people are doing these kind of molecular studies of seeing regulatory changes that could lead to adaptations, or in this case, be selected on by breeders, versus protein coding evolution.
Yeah.
And so it's kind of interesting to see this kind of playing out, I guess, on both sides of the ledger. Here you're seeing regulatory, whereas that paper you mentioned before on headcrest was actually a coding change. Yeah.
Correct. Yeah, and in the case of pigmentation differences, we're actually seeing both, interestingly. So there's been a theme that's emerged from this literature that you mentioned, is that pigmentation genes to some extent are immune from this this problem of deleterious pleiotropic effects. There are a number of different genes involved in generating melanin or pheomelanin that don't really, we think, do a whole lot else. And so in cases of pigment type evolution, there are a number of cases in the literature that we can point to that actually are changes to coding regions of genes that actually do affect the function of those genes or their protein products. And they don't appear to have widespread pleiotropic effects because maybe they don't really do much else. However, in our study of pigeon pigmentation, that turned out not always to be the case. One of the genes that controls a major switch in pigmentation type is a gene called SOX10, another transcription factor that's important in a lot of different processes, including migration of a group of cells in vertebrates that are critical for a number of different developmental processes. That group of cells, the neural crest, gives rise to the face of vertebrates, including ourselves, gives rise to melanocytes, the pigment-generating cells. And the changes that we detected in SOX10 didn't eliminate the function of the gene entirely and didn't change at all the coding region of the gene, but rather they were deletions of a particular stretch of DNA near the gene that just eliminated its expression in melanocytes.
Yeah, that's cool. So it's kind of regulation of regulators in a sense.
Yeah, exactly.
So PIDX1, it turns out, is only half the story, it sounds like. So there's this other transcription factor, TBX5. And so how did that kind of come into the story?
Yeah, so we saw under the other peak of differentiation that we talked about earlier was this gene TBX5. And contrary to what we saw, or in contrast to what we saw with PIDX1, remember the level of PIDX1 went down the bigger the muffs were. With TBX5, expression went up. And the really crazy part about this is that TBX5 was even there in the first place. So TBX5 is another transcription factor from the T-box family of transcription factors. And it's only expressed in the forelimb in most embryos. It shouldn't even be in the hind limb at all. In humans, if TBX5 incurs mutations, especially in the coding region, it leads to a condition called Holt-Oram syndrome. Holt-Oram syndrome affects the forelimb. it also affects heart development. So that's another case of a deleterious pleiotropic effect where one structure is affected, another structure is affected too, that can have some really profound deleterious effects on the organism. But in the case of TBX5, we think the same thing was happening as with PIDX1. Instead of the coding region of the gene being affected, what we see instead is that it's turned on in an abnormal way. It's turned on in the hind limb rather than in the forelimb. And we think the net result of this is that the hind limb, takes on forelimb-like characteristics. So the overall sort of molecular landscape, at least at the level of these master regulators of forelimb and hind limb outgrowth and identity, is that the hind limb identity of the hind limb is actually decreasing with a dialing down of PIDX1 expression. And the forelimb-like identity of the hind limb is being ramped up with this abnormal expression of TBX5, which again shouldn't even be there. It should only be in the forelimb.
So something's controlling PIDX and TB5, which are in turn controlling other things, probably structural proteins, right? And you don't know what they are.
Yeah, eventually structural proteins. So we do know, and not from our work, from work of lots of other people, we do know a bit about the structural protein outcomes, what makes a feather structurally and what makes a scale structurally. But between those 2 points, we know surprisingly little. And that's something that we're currently pursuing is what are those gaps? What happens between the time the limb knows it's a forelimb or a hind limb and those epidermal outcomes are determined? Yeah. That's something we know a lot less about.
So this is worth emphasizing. So you have different birds with or without feathers, and it all comes down to a regulator, not an actual protein making up the cell, which is really remarkable, right?
Yeah, I mean, these are regulators of lots of downstream activities. Yeah. You set in motion, we think, a program that tells particular parts of developing limbs to be either forelimbs or hind limbs. And interestingly, in the case of TBX5, we only see that improper expression in certain parts of the hind limb. It's not expressed throughout the embryonic hind limb. It's primarily at that posterior and dorsal part of the hind limb bud, which is where those big feathers end up growing out. So we think this is not just a partial transformation, but it's also a partial transformation of just part of the limb. Right. So it's having very regionalized effects, which is not something we typically think of when we think of TBX5. We usually think of that as more of a master regulator of outgrowth and identity of the limb rather than something that can have very localized effects.
Yeah, it's really interesting. So then, Mike, if you then kind of go back from the QTL analysis and narrowing it down to these two things, and then look at the sort of pigeon breeds again and correlate these genotypes to phenotype, is it the case that in these muffed guys or the feathered guys that you end up seeing alterations in both of these, of PIDX1 and TBX5, or is it one or the other? Or how does that play out, I guess, in the diversity of the breeds?
Yeah, so we have a limited number of breeds completely sequenced, but what we can ascertain from that is the breeds with very small feathers on their feet, and that's the grouse phenotype you alluded to earlier. Those we think just have the derived version of PIDX1 and not the weird version of TBX5. So it looks like PIDX1 alone is sufficient to put little feathers on the feet. With the ones with bigger muffs, those all have the TBX5 mutation in addition to that. And this, again, is perfectly consistent with what the breeders have known for decades or longer, that it's the two of these things together that act synergistically to make the really big foot feathers.
I see. So far, we've really been focusing on these feathers, but you also, later in the paper, do sort of a really careful job of figuring out the muscle and bone differences in these guys. And so I'm wondering, first of all, what are those differences? And does this represent sort of a trade-off? Like, are these pigeons sort of really bad at walking or moving because they have really weird muscles now? But
yeah, so this part of the study was done in collaboration with one of your departmental colleagues, Gabrielle Cardin, who is also herself a recovering paleontologist. I'm not sure she would want it to be put that way, but I'll do that. Right. So Gab is an expert. I mean, probably one of the world's experts on avian hind limb muscle anatomy. What we know from genetic studies of mice is that if you mess around with TBX genes, some strange things start to happen, not necessarily to the bones, but to the muscles, at least in mice. And one of those things is that the muscles tend to split ectopically. They split a lot more than they should in a normal mouse. So this clued us in that maybe we should be looking not just at the epidermis, but also at what the muscles are doing. And what we found through careful dissections is that this indeed what was happening in a couple of places. So one thing that we observed through these dissections is that a flexor, a muscle that would cause the second toe of the foot to curl if you were to, or the bird was to contract it, actually went to the wrong toe. Instead of going to digit 2, it went to digit 3. So that was unusual. But something that was really unusual is that a muscle called the Fibularis Longus, which is supposed to wrap around the back of the ankle and attach on the bottom of the foot, so that would allow you to sort of extend your toes out or point your foot, instead sent one tendon to the bottom of the foot, but another tendon to the top of the foot, which is basically opposing the action of the other tendon. So when this muscle is contracted, one prediction is that it'll both flex and extend the same joint. So your question about whether that has, whether there's a trade-off involved in locomotion, we've noticed, as has every pigeon breeder everywhere, that some of these big muffed birds definitely walk kind of funny. And it's not entirely clear if they walk kind of funny because they've got 8-inch feathers sticking out of their toes, or maybe now we can hypothesize it has something to do potentially with the fact that their muscles are hooked up in a very strange way. So one way to potentially test this is to study some of these muffed breeds and then start snipping their feathers very early on after they're hatched and see if they still have a strange pattern of locomotion.
So you're wondering if these musculoskeletal differences have anything to do with the feathers in some way, right?
Right. I mean, so one hypothesis would be that if there is a partial transformation from a hind limb-like to forelimb-like identity, certainly we see evidence of that at the epidermal level, but we also see some other changes that are, as we put in the paper, more than skin deep, that perhaps the splitting of the muscles is also a consequence of this misexpression of TBX5. And Nels brought up also the skeletal side of things. We see in some of these breeds a much more elaborated fibula, the bone that's on the outer part of your shin. Most pigeons just have a little splint-like fibula that doesn't even go all the way down to the ankle. So in most vertebrates or tetrapods, including us, the fibula goes all the way from the knee down to the ankle. But in a lot of birds, it sort of tapers off into nothing and never quite gets to the ankle. But in some of these heavily muffed birds, and one breed in particular, the English trumpeter, it's got a really massive fibula. And it goes all the way to the ankle, and it's very robust, which is highly unusual. What it does look a little bit like, and I'm not going to claim that this looks like the corresponding bone in the forelimb, but it's very much like the ulna, the corresponding bone in the forelimb, in that it goes all the way from the knee to the ankle, just like the ulna goes from the elbow to the wrist. But it doesn't resemble the ulna in its morphology.
So these changes are not needed to have bigger feathers, but they are another consequence of the differential regulation of the transcription factors, right?
That's what we think. And what we would like to do next, so we've got all these birds in the cross that we measured for feather morphology, but what we haven't done yet is measured muscle morphology and skeletal morphology. So what we should be able to do is dissect out genetically whether PIDX1 or TBX5 or perhaps something else that we haven't detected yet are the major player or players in determining the muscle phenotypes as well as the skeletal phenotypes.
Now, I think you mentioned earlier something about chickens. You find these genes in chickens as well, and in other birds, maybe?
Yeah. So I mean, the same genes, of course, are in every bird, and in every vertebrate. But what we detected is that in this particular study is that the TBX5 gene that turns on improperly in did you guys hear that ding? Sorry, my computer's making weird.
No, we didn't.
Oh, okay, good. Okay. I'll report next time. sure you'll edit that out. What we find is that TBX5, the gene that is improperly expressed in the hind limbs of pigeons with feathered feet, is also improperly expressed in the hind limbs of chickens with feathered feet. We looked at two different chicken breeds, the Cochin and the Silky, that both have feathered feet. TBX5 is expressed improperly in both of them. We didn't, interestingly enough, we didn't see any changes in PIDX1 expression in either of those different breeds. It's possible that PIDX1 doesn't play the same sort of role as it does in pigeons. It's also possible we simply looked at the wrong stages of development.
It's really cool, Mike. So this idea of kind of converging on the same modifications in TBX5, I mean, what does this say about the mutations that are circulating in these breeding populations? Is it kind of saturating, or, to see the same thing, the same explanation independently? What's going on there? Are there just sort of a limited number of changes you can make to get these sorts of phenotypes?
Yeah, I think there's a couple factors. I think one is exactly that. There's a limited number of things that potentially could be changed to generate the types of traits that we see. So as I mentioned earlier with PIDX1, there aren't that many things that are just expressed in the hind limb that you can envision changes to produce hind limb exclusive phenotypes. And likewise, perhaps there aren't that many things that could be misexpressed in order to give a forelimb-like phenotype in the hind limb. And TBX5 is among the small number of things that do fit that bill. So there's the developmental constraint side of it, meaning there just aren't that many things that can potentially have this effect. Then the other thing you alluded to, which is things that are circulating throughout the population. Certainly, if something has already appeared in a population, that makes it a lot easier to introgress into a new breed. So if somebody with a feather-footed breed encountered another person breeding pigeons hundreds of years ago that didn't have feathered feet, he could have borrowed that bird, crossed it into his breed, and then back-crossed it to his own breed over and over again to get rid of all the undesirable traits of that feather-footed breed except for the feathered feet. Now, that second pigeon breeder is left with his favorite breed, but now it's just got feathered feet, and it's got the genetic scar that came from that first breed that was introgressed into it. So we know this is happening, and that certainly is at least part of the story with feathered feet, because we see the same version of TBX5 and PIDX1 over and over again in different breeds. We know this happens in the wild too. So going back to my other favorite example of sticklebacks, most stickleback populations that live in fresh water have very little armor. So the ancestral stickleback and the sticklebacks that still live in the oceans today have lots of bony armor on the sides of their bodies. But when they move into fresh water, they lose a lot of it. And it turns out the same gene is responsible for that loss just about every time. But it's not just the same gene, it's the same version of the gene that keeps getting passed around from population to population like a genetic disease almost. So certainly sort of a historical precedent, I think, does play a role. Once a particular allele shows up and it's highly advantageous, it can be selected on over and over again by different populations if it's able to make its way in there, either by gene flow or by intentional introgression in the case of domestic species.
Yeah, that's really interesting. So, and this is kind of a total side note, but I'm curious about, you brought up the Silky breed of chickens. Vincent, have you seen these kind of chickens before?
I must have, yeah. It does bring up an image of long hair, right?
Yeah, they just look like they have fur, basically.
Yeah.
I'm I'm dabbling in the chicken hobby these days, and so I'm getting interested. But so, is it known why these silky feathers are so furry in appearance?
So honestly, I don't remember if the mutation has been found. There have been QTL studies that have narrowed it down to particular regions of the genome, but what I don't remember is if that's been taken to the next level to actually identify molecular lesions or changes responsible for it. Interestingly, the silkies have a bunch of weird stuff going on. So they've got feathered feet, they've got the silky feathers. There's also a high prevalence of polydactyly in certain populations.
Okay.
And all of these things appear to be controlled independently. So it's not a single mutation making the feathers silky and the feathers on the feet and too many toes. It looks like everything is segregating independently.
So you mentioned, I think earlier, that in mice TBX5, if you mess with TBX5, you mess up limb development. Is that right? Or was it PIT? PIDX?
Yeah, well, both of them, really.
Both of them?
Yeah, yeah.
So all mammals, all vertebrates have these two transcription factors, correct?
Right, or some version thereof, yeah.
And they're involved in limb development, right?
Yes.
Now, so here we see in pigeons, they're also involved in feather versus scale. So in other, I mean, there may not be an answer to this, but I'm just curious. So here we have an example of a transcription factors that have evolved to have other functions as well. Do we have any other like that in other vertebrates?
Transcription factors that have been sort of co-opted for other...
Yeah, so they have one general function like limb development here, and then in different species, they've been co-opted for other things. Here, feather versus scales, is that a unique...
Right. Well, I mean, in a sense, it's really fulfilling its ancestral function in playing a role in forelimb identity. It's just that it's been... the derived part of this story is that it's expressed in a place that it isn't ordinarily expressed.
Right.
So there are plenty of examples of things being of genes and really whole networks of genes being redeployed in novel context. And another example of that would be development of individual feathers. So feathers are novel structures in the context of vertebrates in general, yet the way that they're specified, front versus back and proximal versus distal, utilizes, at least in part, a very ancient genetic network and interplay between a gene called sonic hedgehog and its downstream targets and bone morphogenetic proteins and its downstream targets. So these same, what we often call genetic modules, have been redeployed over and over again in novel contexts in lots and lots of cases.
Yeah, that makes me think of maybe a related experiment in a way, but it's sort of a reversal of what you observe kind of naturally or in these breeding populations. But I'm wondering, is it possible to think about, altering PIDX1 and TBX5 actually in the forelimbs to sort of do the reverse, where instead of making scaly things wingy, where you make wings into scales? Is that even a possibility?
So that we haven't tried. Trying to think of what's been done along those lines. So what you would have to do is overexpress, say, PIDX1 in the forelimb. And that experiment has been done previously. And what resulted was kind of a mess epidermally. They weren't really feathers. They weren't really scales. But I think what you'd have to do is redeploy that whole hind limb development program in the forelimb. And that I wouldn't say that was done. That wasn't really done. So the jury's still out on that one.
Gotcha.
Yeah.
Well, and that might hint at just that there is some more complexity to these systems. And so it sort of limits the playing field of how much you can really alter them or play around with them in depending on what the context is or the setting is developmentally.
Yeah, I think that's true. So in other experiments, there have been misexpression studies of TBX5 in the hind limb of chickens. So people have done artificially what has been done genetically in these different breeds of pigeon. And when you overexpress TBX5 in the hind limb, you do get kind of weird looking feathers on the feet. It also does have some effect on skeletal development, including the enlargement of the fibula and fusion or loss of some of the phalanges, the toe bones. But it's still always recognizable as a leg. So the foot doesn't turn into a wing or the leg doesn't turn into a wing. It's always a partial transformation. So I think you're right. The system is a little rigged, and there I think is a lot of redundancy in these developmental programs such that a leg will always develop almost no matter what you do to it, but it will take on certain aspects of forelimb identity if you really push it in that direction.
I see. So, and then I wanted to kind of circle back on this idea. We talked about, this idea of convergent evolution with the chickens and the pigeons. And one of the fun things about, or interesting things, I guess you might say, about the eLife papers is that you can actually look at some of the reviewer comments and then see how you actually responded to them. And so I did that. You did fine. But it was interesting because I think at least one or more reviewers brought up the idea that you couldn't use the term convergent evolution in the title of the paper. And so I'm wondering what's sort of the basis of that. Is that because when you think about the artificial selection that pigeon breeders have done, that people kind of still don't consider this evolution? Or is it that the process here might actually be sort of divergent instead of independently derived?
So I think there still is some objection to using the term evolution when talking about domestic species, which honestly I don't completely understand. But it was enough of a distraction in the case of this paper that we kind of toned down that language to get the main points across. But to me, evolution is descent with modification in its purest, simplest form, and I think that domestic species fall well under that umbrella. With respect to convergence, I'm not honestly not sure of some of the nuanced meanings of what the reviewers were getting at. It could have been that they don't think with the case of dinosaurs, for example, that the feathered feet that we observe in Microraptor and other dinosaur species were exactly like what is observed in chickens and pigeons. That could have been an objection as well. And I thought that in the case of these reviews, the specific objection, I think we pointed this out in our response, the specific objection wasn't really laid out as clearly as it could have been for us to respond as meaningfully as we could have.
Yeah, it is really interesting with this new kind of era of the open access and then adding more kind of transparency to the review process. It's kind of get a behind the scenes view of how the conversation happened between reviewers and authors of some of these papers.
I agree. And there have been much more aggressive responses to this idea that domestication is not evolution. I mean, one colleague, who I won't name publicly, has said basically people who believe that must be creationists. They must think that there's something special about humans, that when humans interfere or humans play a role in a process, that we're somehow different than the rest of the world. I wouldn't necessarily go quite that far, but that sentiment is out there.
I think the idea of putting reviews online is good because it gets out of the hidden, nasty comments that reviewers can often make.
Yeah.
I mean, I've seen so many of them over the years. This work is not earth-shattering. That kind of crap.
Yeah, right.
And if you have to sign it, you're not going to say stuff like that.
Yeah, a little sunlight on the process.
One of my colleagues here,
it's great.
One of my colleagues here used to say that the only function of review is to delay publication of your competitor's work, which is not always true, of course, but this could eliminate it. So, Mike, did you ever take this paper and go back to the breeders and show them?
This one in particular, I have sent around. I mean, certainly whenever we publish something on pigeons, I send it around to as many breeders as I am regularly communicating with.
Yeah.
And we often get feedback. In fact, the first couple papers, I had some of the pigeon breeders read it before we submitted it to make sure we didn't royally screw something up on the pigeon side of things.
Probably more than they ever imagined, right?
Yeah, the level of interest is highly variable. There are some people who see it as sort of quaint and tolerate it, and others are really, really excited, and they just can't get enough of it.
It's funny that you would say whole genome sequencing of, 40 strains is quaint.
Yeah. Well, it's just, again, it goes back to our encounter at the state fair several years ago.
Yeah.
If I can't use it to show a bird, it's not that interesting in my everyday life. And I totally understand that. I think if we made this a little bit more practical for them, so if we pitch this as marker-assisted breeding, which at this point we could in some cases, then perhaps there would be a little bit more interest.
No, maybe.
On the other hand, I think part of the challenge for the pigeon breeders is it's funny, sometimes it's like they don't even want to know what's in the background of their birds. They'll acquire a bird at a show. They have every opportunity to know which alleles are hiding in the background for color and other traits. But it's almost like they don't even want to know. They just want to do their own test crosses and figure it out themselves. I think it's part of the challenge.
That's interesting. It reminds me of a paper that came out a couple of years ago. There are chicken breeders around the world who breed chickens who lay blue eggs. And it turns out what they're breeding is a retroviral insertion that went upstream of a blue pigment gene. Right?
Oh, yeah, that's right. Yep.
It's so cool.
It's so cool. I have to say, Vincent, we actually have some Orpington chickens at a community garden about a mile from here that lay some of those blue eggs. And I had forgotten that retroviral connection.
It's very cool.
Yeah.
I love it.
Oh, go ahead.
Go ahead. No, go.
No, I was going to tell Nels a little. I told Nels prior a little secret. If you want some truly spectacular eggs, you can take those blue eggs or the blue egg laying chickens, feed them hot red peppers, and the yolks will turn red.
Wow.
They won't be spicy, unfortunately, but the yolks will turn red. So you can have red yolked, blue shelled eggs.
Yeah, that's perfect. Just in time for Easter.
Exactly.
Cool. That's pretty cool. Pretty cool. Are you still doing pigeon work, Mike?
Absolutely. Yeah, we've got a lot more to come.
Yeah, so you mentioned a couple of traits, the pigmentation traits. What else is on the horizon?
There's still quite a bit more to do for pigmentation. We have addressed some of the big issues in the type of pigmentation that's produced, the eumelanins, the browns and blacks versus the pheomelanins, the yellows and reds. But considerably less is known about how those pigments get distributed. So patterning is a pigmentation patterning in general is a huge issue in vertebrate genetics and diversity. So that's something that we're currently working on. Some of those pigment mutants also have some very interesting pleiotropic effects on eye development, and that's something else that we're getting some traction on as well. Craniofacial diversity still is high on our list of pursuits. So there's a lot more to do. I mean, these birds differ in a lot of different traits. There's also behavioral traits, which I wish we had some visuals on this. Maybe we can have some links from the podcast website to videos of roller pigeons. So these are pigeons that do backflips instead of flying.
Yeah,
they do backflip after backflip on the ground.
Get us the links. We'll put them up for sure.
Yeah, over
That's
and
cool.
over again.
So is your lab all pigeon now?
We're pretty much 100% pigeon at this point, yeah.
Tell me, how many labs in the world work on pigeons?
Well,
any idea?
It depends on how you define work on pigeons. So there are labs that work on pigeon behavior, pigeon navigation, selection on feral pigeons out in the wild or in cities. So there are a number of labs that do that sort of work. I think the number that do the sort of work we're doing is quite a bit smaller.
Yeah. I have to say, the next time I see a pigeon, which will be probably tonight here, I will look at it in a different way after hearing this story.
Much appreciation to the pigeons. You will see the feathered feet out in feral pigeons out in the streets.
Yeah.
They're not that common, but you do see them. So it must not be a huge disadvantage to have them, although we don't know how long those birds survive. But we do see feather-footed pigeons out on the streets occasionally.
Yeah, I'll let you know if I see one.
Yeah. People send me pictures all the time.
I'll be looking for them here, that's for sure.
Cool.
Anything else from you, Nels?
I think that covers it on the paper. This is really fun. We'll get some of these, pictures and images of pigeons in addition to the video of the rollers, just the morphology. It's really fun to see what breeders have done.
Yeah, and I think you said you might be mentioning this, but the Genetic Science Learning Center site has a lot of material on phenotypic variation in pigeons and we can certainly point people towards that.
Yeah, cool. We'll add that as your pick of the week in a minute or two here.
Okay, cool.
All right. So we do picks of the week on some of our podcasts. And Mike, I understand you have something you want to tell us about.
Sure. Do you want me to talk about the GSLC site, or did you want to mention that?
Yeah, do it.
Sure. So as part of our research grant with the National Science Foundation, we have a pretty major outreach component that we've done in collaboration with the Genetic Science Learning Center here at the University of Utah. This is the oldest genetics website, education website on the internet. It's been around since, I think, 1994 or 1996. It's in the top 0.1% of most viewed internet sites. And they've got some amazing teaching materials for genetics and other aspects of biology. In collaboration with them, we've developed a video game called Pigeonetics. So if any of the stuff you've been hearing over the course of this podcast sound interesting to you, you can breed pigeons in the comfort of your own home, on your own laptop, or even on your iPad. without the burden of having to clean up after them. So the game is a lot of fun. There's 26 levels. It actually gets very hard towards the end. And we've also got a bunch of material on the site just discussing variation in pigeons in general. And the nice thing about the game is not only do you learn a bit about rules of Mendelian inheritance in the context of pigeons, but it also links out to some of the molecular work that we've done and some of the work that we've been discussing over the last hour or so. So you can learn more about the molecular side of things in addition to the Mendelian genetic side of things.
So should mention, as you said, this whole site, I guess it's Learn Genetics, right? It's got tons of other stuff.
Right. learn.genetics.utah.edu. And if you go to the home page, I think it's still at the top of the page.
Yep.
There's a banner that says something like genetics at work, and there's a big picture of a pigeon there.
A lot of amazing stuff here. Yeah, I've used these over the years for various other things. I didn't realize there was a pigeon component.
Yes, and there's also a stickleback game.
That's great.
So this is the work of one of my colleagues here in human genetics, Louisa Stark, who's oversees the Learn Genetics site and administers it. So I also added to Mike's. I ambushed him with one or two more picks. So one is the Natural History Museum here at Utah, and it's been really fun because they've actually had an incredible pigeon exhibit, which is ongoing, featuring the Shapiro lab. And Mike is down there on weekends, kind of pressing the flesh and introducing people to his passion for pigeon genetics. So that's another link that we'll put up.
Nice.
And is that still going, Mike?
That goes through early May, I believe. But I'll warn you that the shot glasses are almost sold out in the gift shops. You might want to get there soon.
Yeah, pigeon shot glasses.
A lot of good pigeon swag in the gift shop.
I bet you have some of that, right, Mike?
I've probably bought about half of them at this point. Yeah, they make great gifts.
And then I have one other surrogate pick for Mike, which is this great new book by Richard Bailey. I believe it's called Darwin's Pigeons, with these amazing photographs of, again, a lot of this diversity of the breeds.
Yeah, it's pretty cool. A nice picture on the cover.
Yep.
Really amazing.
Yeah, and you'll see banners of some of those in the museum exhibit.
Nice. Nels, what do you have?
Well, I've, we've been doing a lot of kind of winged creatures in TWiEVO, and so I wanted to start maybe pushing us in some new directions. And I've had parasites on my mind for a while. So I might have mentioned it. I was just at a meeting helping organize a symposium on microbial control of host behavior. And so one of the videos shown by one of the speakers at this meeting, this is a fellow called David Hughes, was a striking video of these parasitic hairworms that infect crickets, for example, and actually are mind-altering parasites that cause the crickets to jump into water when they otherwise wouldn't. And this sort of promotes the replication cycle of these hairworms, and they sort of weasel their ways out of the crickets. It's really just kind of terrifying and disgusting. So anyway, inspired by that, I found, I was just kind of reading the Washington Post the other day, and they have a little piece that I have a link to here on the emerging National Parasite Collection, which is coming to the Smithsonian Museum. And it's still, I think, to be determined when this exhibit will emerge or how exactly it will happen. But it's pretty crazy. They've something like 20 million parasites in jars. These collections have been around for years, I guess, just sort of out of the public view. And so I'm at, some great just drawings and ideas as this comes together. So something to keep an eye on will be this National Parasite Collection.
Got to check that out for sure.
Yeah.
But, how about you? So if you should visit in April, Nels, my colleague Dixon Despommier has a little collection here outside.
Oh, of course. Yeah. Actually, this is kind of speaking of parasitic control of host behavior, I was just wondering about your colleague Dixon. Maybe he's infected with some parasite that could explain his passion for all of the, collecting, keeping, studying, inspiring others.
One might argue that we all have some kind of parasite that drives us, right?
That's right.
Who knows?
Yeah. How about you, Vincent? What's your pick?
So I have a pick inspired by what I'm going to do tonight. After we're done here, I am heading downtown. We have a well-known bookstore here in New York called the Strand Bookstore. And I'm going to do something called a parlor chat. I was invited by Stuart Firestein, and Stuart told me about a month or two ago, he goes, this guy Ben Lilly is doing these parlor chats, and you should come and we'll just talk about viruses. Firestein, by the way, wrote a very interesting book called Ignorance.
Oh, yeah. I remember reading that actually from a tip from you on TWiEVO.
Yeah, so we had him on TWiEVO a long time ago, and he has a course called Ignorance, and I've participated in it. So he said, you would be good for this because we're going to sit in front of an audience in this bookstore and talk about what we don't know about viruses and so forth. So I looked up this fellow Ben Lilly because I didn't know who he was. And it turns out he's a physicist who got together with another physicist, and they do things to try and bring science to people. They have a podcast called The Story Collider.
Oh, yeah.
And what they do is they have people stand in front of a mic, mostly scientists, and talk about a moment in their life that means something to them. You know, so this is The Story Collider. They have a website where they talk all about this, and they have podcasts as well. So they do a lot of science outreach, and there are lots and lots of these recordings. They're pretty short. They're all less than a half hour of people just talking about, scientists talking about some experience they had that was transformative for them. So I think it's pretty cool. I'm looking forward to meeting Ben Lilly tonight and doing that, but I thought our listeners might like to check this out, storycollider.org.
Yeah, this looks really interesting. So I just pulled it up, and I see right at the top this fellow Joe Pelka, who does a lot of NPR stories on science. I've been a fan of his for quite a while.
Very they've got a number of people you might know. One of them, George Church, is in there.
Oh, sure. Yep.
Talking about various things. So check it out. It's cool.
Yeah, thanks for the tip.
And, again, that's because I'm going down to do something sponsored by Ben Lilly, so should be fun.
Yeah, enjoy.
All right. That should do it for TWiEVO number 7. We're marching towards the magic number 12 now.
We're getting there. It's going to happen.
And then we're going to be home free, right?
That's right.
What happens to 12?
Well, 12 is the magic number for podcasts. Most don't get past number 12.
Oh, you guys got it.
And we will, because we've already done very well. We got a lot of listeners, and I've never started a podcast that I've had to stop.
No, I feel like we have great momentum, and we're just kind of scratching the surface. So
there's
plenty of guests in various areas to explore. So I'm feeling like we're gaining energy.
No, this is great, and this is all stuff that I would never ever read about, and so I really love it because I get to learn about lots of different things. So it's great, and I appreciate your expertise, Nels, at bringing new people to the fold. Like Mike. So thank you.
My pleasure. This has been really fun. Thanks, Mike.
Mike is gone.
Thanks for having me.
Mike is gone.
He's still here.
I'm here.
Mike is a professor at the University of Utah in the Department of Biology. Same department as you, Nels. Are you in genetics? Is that correct?
Well, so I'm in genetics. Mike and I both have adjunct appointments, however, in our sort of respective. So Mike's an adjunct in genetics, and I'm an adjunct in biology. And this kind of reflects, we just have this really fun community where we're kind of exchanging ideas and working together in new and interesting ways. And so Mike and I have had a lot of conversations over beers and lunch and things like that. And we're trying to grow evolutionary genetics here at Utah in new ways.
Wonderful.
And we teach together too.
Exactly.
I hope to meet you when I visit, Mike.
Yeah, likewise.
At some point in the future. Anyway, we thank you a lot for participating. And
my pleasure. Thanks for having me.
Lovely work and lovely pigeons.
Thank you.
Nels Elde is at cellvolution.org. You can also find him on Twitter as Al Earlybird. Thank you, Nels.
Thank you, Vincent. This is fun.
I'm Vincent Racaniello. You can find me at virology.ws. And don't forget, you can find TWiEVO at iTunes and also at microbe.tv/TWiEVO. And send us your questions and comments to TWiEVO at microbe.tv. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.